That finding came from a researcher named Dr. Doron Behar at Rambam Medical Center in Israel. In 2019, his team analyzed 153 unrelated Assyrian participants. What they found stopped the analysis. HV4A2A and its parent group appeared at a frequency of 17.6% in Assyrian samples, the highest ever recorded for this lineage in any sampled population on Earth. And HV4A2A itself, the specific subclade, showed up concentrated in Assyrian communities in a way that made geneticists look twice. The molecular clock put its origin squarely in the Bronze Age Tigris highlands, right where Assyria was. Now stay with me, because this next part is the reason this video exists. This is not a story about an empire that fell. Every history class taught you the Assyrians were conquered, absorbed, erased. Babylon broke Nineveh. The Persians came next, then Alexander, then the Arabs, then the Mongols. Layer after layer, civilization after civilization, rolling over the same ground. And yet, here is a man in Gothenburg carrying a 4,000-year-old maternal lineage that went through all of that, intact, unbroken. The textbook said assimilation. The DNA said something else entirely. His grandmother's name was Miriam. She was 7 years old in 1915 when forces drove the Hakkari Assyrian tribes out of the mountains of southeastern Turkey. 70,000 people. She walked south to the Mesopotamian plains. She survived. She carried HV4A2A in every cell of her body, passed down through her mother and her mother's mother and every mother before that going back 40 centuries. She died in Gothenburg in 2003. She never gave a DNA sample. Or she didn't know the name for what she was carrying. If you've ever wondered where you really come from, and I mean really, then subscribe, because this is only the beginning. The question that nobody has asked out loud is this: what had to happen to a people? What sustained, relentless pressure had to be applied across thousands of years to keep a bloodline this isolated, this intact, this old? The answer is not what you expect, and it starts with a brick. Before we go any further, I want to know something from you. If a DNA test revealed that your family's ancestry traced back to a civilization that history said was completely erased, would you want to know? And more importantly, would it change anything about how you see yourself? Drop your answer in the comments. Yes, no, or something more complicated. I read every single one. And what comes next in this video might actually change your answer before you finish watching. Isolated. That's the word. But isolated in what? In rock, in clay, in a king's brick. Let me tell you about a storage vault at the National Museum of Denmark in Copenhagen. It's 2020. A clay brick sits in a climate-controlled drawer, the size of a shoebox, roughly the weight of a newborn. It's been sitting there for over 100 years, ever since European archaeologists pulled it out of the ground in northern Iraq. A crack had been slowly widening in its lower half. Nobody thought much of it. Then Dr. Sophie Lund Rasmussen from the University of Oxford and her colleague, Dr. Troels Arbøll, looked at what was visible inside that crack and asked a question that nobody had thought to ask in 150 years of Assyrian archaeology. What if the clay preserved DNA? The brick bears a cuneiform inscription in Akkadian. Translated, it reads, "The property of the palace of Ashurnasirpal, king of Assyria." It was made in Kalhu, modern Nimrud in northern Iraq, between 879 and 869 BCE. That date is not an estimate. It's derived directly from the king's inscription. The brick is precisely dated to a 9-year window during the reign of one of the most powerful rulers the ancient world ever produced. Stay with me, because this next part is the reason this video exists. Rasmussen's team drilled into the inner core of the brick, the part least exposed to the outside world since it was fired. They extracted ancient plant DNA. Say for the first time in the history of archaeology, biological material had been successfully pulled from a clay artifact nearly 3,000 years old. What they found inside was 34 distinct groups of plants: cabbage relatives, heather, birch, cultivated grasses, aromatic medicinal herbs, an entire ecological snapshot of a specific garden in a specific palace complex in 875 BCE, Not described, not reconstructed from written records, chemically present, sitting in a museum drawer in Denmark for a century. Nobody looked. Here's the part that gets under your skin. Birch doesn't grow in the Mesopotamian lowlands. It grows in the cooler highland zones of the Zagros Mountains. Someone had to transport those trees, physically move them from the mountains to the palace because a king wanted shade. That took timber workers. His botanical knowledge, long-distance supply networks that left no written record. The people who did that work never made it into the royal annals. Their names are nowhere in the historical record. But their world, the plants they touched, the soil they packed around royal roots, survived inside a brick in Copenhagen for 2,900 years. That brick is their only monument. And if a brick can hold biological memory for nearly 3 millennia, think about what a human genome holds. Dr. Lazaridis at Harvard Medical School had been working on exactly that question. In 2022, his team published the first ancient DNA data ever collected from the Pre-Pottery Neolithic of Mesopotamia, southeastern Turkey and northern Iraq, the precise geographic homeland of the Assyrian people. What they found was a genomic continuum, a biological thread running from 10,000-year-old Tigris Valley farmers all the way through to populations in the same region today, unbroken. The gene pool changed, yes, admixture happened, migrations happened, but the core Mesopotamian signature never disappeared. And yet, buried in the supplementary data was something that didn't fit. A Y-chromosome lineage appearing in living Assyrian communities at frequencies that had no explanation in the ancient Mesopotamian record. A lineage that, by all the rules of paleogenomics, should not have been there. Statistically impossible, paleogenomically anomalous. The date was wrong, the result was real. Impossible is a word that survives exactly until you find the Hurrians. Picture this, the Pontic steppe around 2900 BCE. A man is buried in a mound near the lower Volga River. When his Y-chromosome haplogroup is R1b-Z2103, the dominant paternal marker of the Yamnaya people, the Bronze Age steppe herders, whose migrations reshaped the genetics of half the world. His descendants will ride west into the Balkans. They'll push south through the Caucasus. And somehow, through a chain of events that geneticists are still trying to reconstruct, that same bloodline will end up inside Assyrian communities in northern Iraq and Iran, not at trace levels, not as a footnote. In some Iranian Assyrian subgroups, that Y-chromosome haplogroup reaches 40%, higher than in Armenia, higher than in Azerbaijan, higher than in any population geographically between the Volga and the Tigris. Nobody has explained it. Dr. Siiri Rootsi at the University of Tartu in Estonia spent years mapping Y-chromosome distribution across the Near East. In 2006, her team published a study comparing Assyrians, Syrians, Kurds, and other groups in the same region. What they found was stark. Assyrians and their Syrian neighbors, two groups speaking related Semitic languages, living in the same cities for over 1,000 years, were very distinct from each other at the Y-chromosome level. The Assyrian paternal profile had no close parallel in any geographically adjacent population, and one of the lineages driving that distinctiveness was the steppe-linked R1b cluster that shouldn't have been there. But here's what changed everything we thought we knew about how empires absorb the people they conquer, the Hurrians. They entered northern Mesopotamia from the southern Caucasus foothills somewhere between 2300 and 1500 BCE. They spoke a language with no known relatives, not Semitic, not Indo-European, something entirely its own. They brought horse-drawn chariots, bronze metallurgy, and a Y-chromosome pattern consistent with people who had come through the Caucasus corridor, the same corridor that connected the steppe world to the Near East. They built a kingdom called Mitanni, directly adjacent to early Assyrian territory. They intermarried with Assyrian elite families, and then, around 1340 BCE, Mitanni collapsed under combined Hittite and Assyrian military pressure. The Hurrian state was gone, but the Hurrian people weren't destroyed. They were absorbed by the very empire that crushed them. Their Y-chromosome went with them. So there is a letter in the British Museum, one of the Amarna tablets, written around 1350 BCE, from the Mitanni king Tushratta to Pharaoh Amenhotep III of Egypt. He writes in Akkadian, the international diplomatic language of the Bronze Age. He calls the pharaoh, "my brother." He sends gold, lapis lazuli. his own daughter, Tadukippa, as a political bride. Within 40 years of that letter, Tushratta is dead, his kingdom is erased, and his people's paternal lineage is being folded into the Assyrian gene pool. The R1b-Z2103 signal in Iranian Assyrians today, at 40%, may be the direct biological inheritance of Tushratta's bloodline, carried forward 3,400 years in a population that barely changed around it. So here's what the Assyrian genome actually is. It's not a simple Semitic lineage, it's a layered archive. Neolithic Tigris farmers at the base, the maternal HV lineages going back 10,000 years, then Levantine Semitic populations moving north, then Hurrian steppe-linked warriors absorbed after 1340 BCE. 3 genetic worlds fused under one identity, and that layered structure, that specific combination, is exactly what made Assyrians genetically distinct from every neighboring group, and exactly what made them impossible to dissolve. But here's where it gets strange, because the people who came closest to dissolving them weren't the Babylonians, they weren't the Mongols, they were a 20th century state with a modern army and a policy, and the evidence for that is not in a history book, it's in a genetics paper, in a single word buried in the methods section, a word most readers skip: bottleneck. A bottleneck in population genetics is not a metaphor, it is a body count. The year is 2013. A population genetics study examining Assyrian highland communities from the Hakkari mountain region of southeastern Turkey has just been published. Buried in the methods section, noted without elaboration, is a finding that the research team marks as demographically significant. The Hakkari Assyrian subgroup shows a pattern of genetic diversity consistent with a recent severe reduction in effective population size, a sharp contraction. The clinical word for it is bottleneck. The historical word for what caused it does not appear in the paper, but it has a name: the Sayfo. In Syriac, the ancient language these communities still speak, it means the sword. Between 1914 and 1923, while the Ottoman forces and Kurdish irregular militias killed an estimated 155,000 to 300,000 Assyrian Christians across southeastern Anatolia, the Hakkari Mountains, and northwestern Persia. The Hakkari Assyrian tribes, genetically the most isolated, the most ancestrally preserved, the most concentrated holders of pre-modern Near Eastern lineages anywhere in the world, were driven from their mountains in a military campaign that began in September 1915. The families that had been living in those highlands for thousands of years, the people whose genes connect most directly to those Neolithic Tigris farmers, were shattered in a single mountain season. This isn't just ancient history. The erasure is still happening, in databases you can look up right now. Dr. Sargon Donabed at Roger Williams University in the United States has spent his career documenting what happened next, not just the physical destruction, but the administrative erasure that followed it. Assyrian survivors who made it to the United States were classified at Ellis Island as Syrians, Persians, or Turks, whichever Ottoman province they had fled from. The genocide didn't only kill people, it reclassified the survivors. It removed the name from the record. The paper trail that states used to define a people's existence was quietly, systematically rewritten. Actually, if you watched my last video on why Iranian DNA is so ancient, you already saw the first half of this puzzle. The population isolation we documented there, this is what creates it. Not just geography, something more violent than geography. What did you think of that video? Did it change how you see the region? Drop it in the comments, and then stay with me. Because what comes next is what that video was building toward. There are 3 things the 2013 bottleneck study revealed. The third one is the one nobody talks about. The HV4A2, a lineage is passed only through mothers. It has a molecular clock age of 3,300 to 4,200 years. Every woman alive today who carries it is a direct link in a maternal chain that ran unbroken from Bronze Age Mesopotamia to the 20th century. The rarity of the lineage today is not only a function of ancient population dynamics. It is, in part, a count of how many of its carriers were killed between 1914 and 1923. The frequency of an ancient bloodline is also a ledger of a modern atrocity. And here is the third thing. The Hakkari Assyrian population was already showing signs of extreme reproductive closure, endogamy, marrying only within the community, before 1914. The isolation preceded the genocide. The genome had been defending itself for centuries before the military campaign began. Multiple demographic stress events, the researchers noted. Not one catastrophe, a pattern. Something, not a single army, but a sustained pressure across generations, had been narrowing this bloodline long before the Sayfo formalized the attempt. A 4,000-year-old bloodline, a centuries-long pattern of biological siege, and a genocide that nearly completed it. The question that follows from all of that is almost too uncomfortable to ask. What exactly was being protected, and from what, for all those centuries before 1914? Centuries of pressure do something strange to a genome. They turn it into a wall. Northern Syria, 2008. A population genetics study samples ethnic communities across Iraq: Arabs, Kurds, Turkmen, Yazidis, Assyrians, Mandeans. The researchers calculate genetic distances between every group. When the Assyrian cluster appears on the analysis, it doesn't sit near its geographic neighbors. It sits alone. The genetic distance between Assyrians and Arab Iraqis living in adjacent city neighborhoods is larger, statistically, than the distance between populations separated by the Sahara Desert. 1,400 years of shared geography, zero genetic convergence. Dr. M.T. Akbari at the University of Tehran ran that analysis. Her team was looking for evidence of population mixing in one of the most ethnically diverse regions on Earth. So she found mixing everywhere, except in one community. The Assyrian samples simply didn't move toward any neighboring group. No drift, no convergence. Genetically, they were an island. Then, in 2011, Dr. Nadia Al-Zaheri at the Italian National Research Council published her landmark study, a survey of Y-chromosome and mitochondrial DNA variation across multiple Mesopotamian communities, including Marsh Arabs, Assyrians, and Mandeans. Al-Zaheri spent years on that question. What her data confirmed was that all three groups carried DNA that was predominantly of autochthonous Mesopotamian origin, meaning it came from people who had been there in that specific river valley for a very long time. Not from Arabian expansion, not from Iranian migration, from the ground beneath their feet. This finding was published in 2011. It's been cited 92 times. The mainstream history curriculum has not changed a single sentence. So, what built the wall? How does a community living surrounded by other communities for 14 centuries share essentially no genetic material with any of them? The answer is not geography. Assyrian communities in Baghdad, in Urmia, in Damascus were equally isolated genetically. The wall was not made of mountains. It was made of a single Greek word. In 431 CE, at the Council of Ephesus, the Patriarch of Constantinople, a man named Nestorius, was condemned as a heretic. His crime was teaching that Mary was the mother of the human Christ, not of God. One word in Greek, Theotokos, God-bearer, and whether it applied to her or not. Nestorius said it didn't. The council said it did. He was exiled. His followers fled east. And they built the Church of the East, what history calls the Nestorian Church, and they established communities in northern Mesopotamia, Persia, India, and eventually as far as Tang Dynasty China, the most expansive missionary network of the pre-modern world. And in every community they planted, the rule was the same. You marry within the faith. That wall went up in the 5th century. It didn't come down. But here's where it gets strange, because the endogamy, the closed reproduction, was already present in Assyrian communities before Christianity formalized it. Rootsi's 2006 Y-chromosome data and Al-Zaheri's 2011 mtDNA survey both point to a pattern of reproductive closure that predates the Council of Ephesus. Christianity didn't create the wall. It gave the wall a name, a doctrine, and an institution. In something older, something that looked like sustained existential pressure across generations, had already made isolation a reflex. Christianity made it a rule, and buried in all of that, between the Hurrian absorption of 1340 BCE and the rise of the Nestorian Church in 431 CE, there is a haplogroup cluster in Assyrian mitochondrial DNA that no peer-reviewed study has formally named or identified. Lineages consistent with a Bronze Age movement from the Aegean or Anatolian coast. Who these people were, when they arrived, what they called themselves, unknown. They left descendants in every Assyrian community on Earth. They left no record, and the highest concentration of their signature is not in Iraq or Turkey or Iran. It is on an island in the Aegean, an island that no one has ever connected to Mesopotamia. That island is Crete, and what the Minoans were carrying in their bones was not European. In 2013, researchers analyzed ancient skeletal remains from a Bronze Age ossuary at Agia Triada on Crete. The Minoans, the people who built Knossos, who gave Bronze Age Europe its maritime trade networks and its bull-leaping art, had never been genetically characterized in 150 years of serious archaeological study. The team expected to find European hunter-gatherer ancestry. They found a haplogroup HV in Crete, in Bronze Age bones, at nearly 9% frequency, described in the analysis as more typical of Mesopotamia than of anywhere in Europe. Additional haplogroups in the Minoan samples, R0, H5, H7, H13, A1A, were all lineages associated with Bronze Age Near Eastern populations. The most straightforward explanation the researchers could offer was a Bronze Age migration of women from the Mesopotamian or Levantine coast into Crete, sometime before 2000 BCE. Everything I just told you about the unnamed lineage in Assyrian DNA, hold on to it, because it's about to mean something completely different. Here's where it looks like the mystery resolves. The Minoan connection explains the unnamed haplogroup cluster. The migration ran from Mesopotamia outward, women carrying HV lineages westward across the Mediterranean, eventually settling in Crete. The unnamed people were Mesopotamian. Their descendants built Knossos. Case closed. Except, and this is where the floor shifts, if the haplogroup left Mesopotamia and went to Crete, then the people who stayed behind in Mesopotamia weren't receiving foreign DNA. One, they were sending it out. The Minoan civilization wasn't influenced by Mesopotamia, it was partially made of it. And two civilizations that every textbook treats as separate, the Minoans of the Aegean and the ancestors of the Assyrians on the Tigris, were telling the same genetic story from opposite ends of the Mediterranean. Think about the palace at Akrotiri on Santorini, destroyed in the Thera eruption of around 1627 BCE. The frescoes there show blue monkeys, monkeys native to Africa, not Greece, lily paintings that match specimens from the Nile Delta, trade goods spanning Egypt, the Levant, Mesopotamia, and the Aegean in a single interconnected Bronze Age commercial world. The HV maternal lineage was the bloodline of the women who lived inside that network, who moved goods, plants, ideas, began DNA across the Mediterranean 2,000 years before Rome. It began in Mesopotamia. It ended in Crete. When Thera erupted and the network collapsed, the haplogroup survived, in the mountains above the Tigris, in communities that never saw the sea. And yet, the political record says the exact opposite of the genetic record, every time. Because in 2008, when geneticists confirmed that Assyrians were genetically distinct from every neighboring population, they created a problem for the states those neighbors belonged to. In Turkey, Assyrian communities are legally classified as Syriac Christians, a religious subcategory that erases ethnic distinction. In Iraq, under the Ba'athist constitution, they were a religious minority, not an ethnic group. In the United States, before 1940, Sayfo survivors arriving at Ellis Island were classified as Syrian, Persian, or Turkish, whichever Ottoman province they had fled. Two independent genetic studies, 20 years apart, confirm the same conclusion. This is a distinct population with an ancient, continuous identity. Every state document says there's something else. The DNA says one thing, the border says another. The question is whether the mismatch was an error or a policy. Policy leaves a paper trail. So does its absence. Pick up your phone, open 23, and me. Open Ancestry, DNA. Go to the reference population section, the list of named ancestries the platform uses to identify where your DNA comes from. Scroll through it. You'll find Yemeni, Jordanian, Iraqi Arab, Kurdish, Azerbaijani. You will not find Assyrian. What's the world's oldest continuously documented civilization? A people whose maternal lineage has been in the same geographic region for potentially 10,000 years, does not exist as a named category in any major consumer genetics platform. If you are Assyrian and you submit your DNA, the algorithm returns something like unassigned Middle Eastern. the most ancient continuous genome in the Near East, labeled as unassigned. This is not a technical oversight. Building a reference population requires samples, funding, institutional interest, and a classification framework that recognizes the group as a distinct entity. None of those exist for Assyrians at scale, because for over a century, every major state in the region has had an active interest in classifying Assyrian communities as something else. Dr. Sargon Donabed at Roger Williams University has documented this specifically. His research traces how Assyrian communities arriving in the United States after the Sayfo were systematically misclassified in immigration records, census documents, and political frameworks across Turkey, Iraq, Iran, and Syria. The paper trail of erasure is meticulous. The paper trail of existence is almost gone. The genetic continuity is 10,000 years old. The official record is less than a century old. And in any court, in any bureaucratic system, the official record wins. But wait, before we get there, there's something the study didn't publish, something that sits in the gap between what the data shows and what anyone has chosen to say about it. In 2017, meets a study published in PLOS 1 examined paternal lineages of multiple ethnic communities in northern Iraq: Assyrians, Syriacs, Turkmens, Yazidis, Arabs. The researchers, led by a team based in Iraq, found that Assyrians and Christian Syriacs, two communities that modern states routinely treat as the same group, were genetically distinct from each other, not slightly, substantially. At the Y-chromosome level, they clustered separately. Two communities legally merged under one administrative label in multiple countries. Genetically, they are not the same people. And here is where the missing variable becomes truly significant. There is no comprehensive comparison between pre-Sayfo Assyrian genetics and modern Assyrian genetics. The baseline doesn't exist. Nobody collected samples from the Hakkari community before 1915, which of the ancient DNA record from Mesopotamia ends before the communities that would become the modern Assyrian diaspora were formed in their current structure. What the genome looked like before the Sayfo is a gap, a specific, permanent, unrecoverable gap in the scientific record. The genocide didn't just kill people, it deleted the control group. It removed the data point that would allow us to measure exactly how much was lost. Someone, somewhere, may have the data to partially reconstruct it. Diaspora communities who emigrated before 1914, particularly the Urmia Assyrians in Iran, identified by researchers in 2000 as the least admixed Assyrian subgroup ever sampled, carry genomes that are the closest living approximation to that baseline. That data exists inside living people right now. As of today, no comprehensive ancient versus modern Assyrian genomic study drawing on those communities has been published. The data is out there. Nobody has released it. Now, I'm going to tell you something that contradicts everything this video has been building, and I'm going to make the case as strongly as I can, because it deserves that. The Assyrian continuity narrative, the idea that modern Assyrians are the direct, unbroken descendants of the ancient Assyrian Empire, is not simply a scientific conclusion. It is also an identity claim. It is used to support demands for political recognition, for territorial autonomy in northern Iraq, for recognition of the Sayfo as genocide. And critics, Arab scholars, Kurdish scholars, Turkish scholars, have pointed out, not unreasonably, that genetic data is being used to make political arguments, that the science is being recruited in service of a cause. And here is the most uncomfortable version of that critique. The Assyrian Empire itself was one of the ancient world's most aggressive practitioners of mass deportation. Under Tiglath-Pileser III, Sargon II, and Ashurbanipal, the Neo-Assyrian Empire deported an estimated 4.5 million people across its territory between 745 and 612 BCE. Entire populations moved. The northern Israelite tribes, deported by Sargon II in 722 BCE, 27,290 people, by his own royal count, inscribed in the annals at Khorsabad, Babylonians, Elamites, mountain peoples from the Zagros. If the Assyrians absorbed all of these populations through forced migration, why is the modern Assyrian genome showing near zero admixture? Shouldn't the empire's own policy have diluted it? That is the strongest version of the counterargument. It deserves a straight answer. The answer is the Church of the East, but not in the way Segment 5 described it. Run the argument in reverse. The Assyrian Empire's mass deportation policy may have accelerated genetic isolation rather than diluting it. A community that watched its own state machinery move millions of strangers through their homeland would have every reason to close its reproductive boundaries early. The endogamy that preserved the genome wasn't just a religious rule applied in 431 CE. It may have begun as a survival behavior in the final centuries of the empire itself, a population learning, before anyone named it as theology, that staying genetically within the group was the only way to stay a group at all. The Assyrians survived their own empire's policy by becoming immune to it. Not through power, they lost power completely, through geography. The Hakkari Highland Corridor, at the intersection of modern Turkey, Iraq, and Iran, with altitude ranges that made occupation expensive and resupply nearly impossible. Every army that conquered Mesopotamia looked at those mountains and decided the cost wasn't worth it. The Persians left them, the Greeks left them, the Arabs left them, the Mongols, who destroyed everything south of the mountains, left them. The Assyrians didn't survive because of civilizational strength. They survived because they lived somewhere that nobody wanted badly enough to pay for. Until 1914. That's the terrifying finding. Not that the Assyrians survived, it's what their survival proves. Every empire for 2,600 years looked at the Hakkari Highlands and chose to leave it alone. The Sayfo was the first time a modern state decided those mountains were worth clearing, and the genome recorded that decision in a bottleneck, in a diversity contraction, in a specific pattern of demographic stress that sits in a peer-reviewed database right now, labeled demographically significant in the methods section of a 2013 paper that almost nobody has read. The language of genocide has been readable for 2,700 years. Sargon II recorded his deportation of 27,290 Israelites with the clinical precision of a logistics report. We have read that inscription. We have translated it. We have put it in museums. We never once treated it as a legal document until 1948, when a Polish lawyer named Raphael Lemkin gave the pattern a name. In the genome is also a document. It also describes a pattern. The question is when we decide it counts as evidence. Chicago, Illinois, 2023. The Assyrian American National Federation publishes a report. Since 2003, the Assyrian population of northern Iraq has declined from approximately 1.5 million to under 300,000, an 80% reduction in 20 years. The same geographic zone where Lazaridis' 2022 science paper identified a 10,000-year genomic continuum. The people leaving on flights from Arbøll to Toronto to Sydney to Stockholm, they are carrying the same genetic signature as the people who built the palace at Kalhu in 879 BCE. The same maternal lineage as the brickmakers whose plant DNA sat in a Copenhagen museum for 150 years. When a family boards that flight, they take the archive with them. The archive survives. See, but the context does not. Think about what a genome can't carry. It carries the haplogroup, it carries the molecular clock age, it carries the bottleneck signal and the endogamy pattern and the Hurrian Y-chromosome and the Minoan maternal connection. But it does not carry the specific sound of a river in the Hakkari Highlands at spring melt. It does not carry the oral tradition in Assyrian Neo-Aramaic, one of the oldest continuously spoken languages in the world, that describes the mountains by names used before Rome existed. It does not carry the agricultural knowledge of people who farmed the same terraces for 100 generations. The DNA is the archive. The library is the land, and the library is burning. There's a man named Asher. You met him at the beginning of this video, kitchen table in Gothenburg, holding a DNA printout. His grandmother Miriam walked out of Hakkari in 1915. His parents left Urmia in 1988. He left northern Iraq in 2004, 3 generations. 3 expulsions from the same region. 1 haplogroup carried through all of it. His children were born in Sweden. They have never seen the Tigris River. They carry HV4A2A. If you want to understand what ancient DNA actually reveals about the civilizations being erased right now, not 3,000 years ago, but today, subscribe, because the next episode goes somewhere colder, older, and far more uncomfortable than anything in this one. Here's what the science cannot tell you. It cannot tell you whether the people being displaced from northern Iraq today are the same people in any meaningful human sense as the ones who built Nineveh. Genetics is not identity. A haplogroup is not a homeland. A molecular clock age is not a deed of ownership. These are things humans decide. In courts, in constitutions, in history books, in the names they choose to give to massacres. The genome doesn't make those decisions. It only records them. And the record shows a population that has been here for 10,000 years, that survived Babylon and Persia and Greece and Rome and the Mongols and 7 centuries of Ottoman rule. That encoded a genocide in its own mitochondria, that carried the oldest maternal lineage in the Near East through every catastrophe the last 4 millennia could produce. And that is right now, at this moment, leaving the only land that lineage has ever known. The genome survived. Whether the people attached to it do, that is not a question science can answer. That is a question we are answering right now in real time, shaped with policy and silence and things we choose not to name. The Sayfo was never formally recognized by most of the world. The second dispersal has no name at all. The bottleneck is still narrowing, and somewhere in a database that almost nobody reads, the signal is there, documented, peer-reviewed, filed under demographically significant, waiting for someone to treat it as what it is.